So you had an announcement with OpenAI around really, it seems like reducing the cost of cell-free protein synthesis.
Yep.
And we'll actually get to see the lab where sort of the AI model was mated with the autonomous lab and where the work got to be done. And I feel like there's an interesting, almost kind of back and forth relationship with when you go into autonomy, which is there's, from the Nature article that I read and so forth about it, a real emphasis on reducing costs in order to do science and be able to do science at scale and be able to look at a much larger, maybe like problem space in order to find interesting solutions. When you do work in the real world, it requires reagents and precursor chemicals and all sorts of very expensive stuff. At the same time, right, it seems like the North Star for this work that you did was to reduce those costs. So in some ways, it's like to do the work requires maybe more cost than usual, but then as you do it, maybe you find efficiencies that otherwise wouldn't have been found. So tell me more about that. It seems like a really interesting dynamic here.
Yeah, so that's a very good insight you had there. Actually, that's one of the reasons we picked that as a target for the project, because we thought it was sort of like AI helping itself. Because one of the big challenges, as you're pointing out, and this is again why the frontier labs are starting with math and physics, is that's free, right? Why did DeepMind start with chess? Right? Like, well, you can play games of chess for nothing, right? And you're on 1 million games of chess on a processor, right? For nothing. And so, yeah, it uses power, but like comparative to using atoms and building things, it's cheap. So absolutely, right? Like now, to be fair, again, the dominant cost today, I cannot emphasize enough in doing the atoms work of biotechnology is humans and physical space, not the reagents. The reagent budget, even at a highly automated place like Ginkgo, like back when I wasn't on the autonomous lab and we were just doing like walk-up automation and work cells, it was like sub 7% of the budget. It's just dominate, like it's just so expensive given relative to the throughput of the people. Okay, so let's just be clear. The actual first problem is remove the vast amounts of like overhead for inefficient lab space, low utilization devices, and humans moving stuff around all that, and let the humans think and not do that manual labor. Okay, that is step one. After you've completed that step, it's still gonna be expensive compared to playing chess. But at least it'll be less expensive than doing it at the bench. Does that make sense? Okay, and then now we're into the era of, oh, it's all running autonomous and it's all about the reagent costs. Why don't we reduce the cost of the reagents? And so that was the project we did with OpenAI. We're like, one of the best fits, we think, for a lot of the AI for bio stuff right now is in just protein design. It's a tractable problem. Proteins are not the infinite complexity of a whole cell. And so starting with AlphaFold at Google, and like Demis won a Nobel Prize for this, but like on to newer models, newer versions of AlphaFold, but companies like Chai Bio and others are basically making models to design proteins against therapeutic targets. Great. How do you make the proteins? Well, you can obviously put it into a cell like a CHO cell, but when you do manufacturing, it's typically done this way. But in the lab to test many proteins, it's often done in a cell-free mix, at least for these AI guys especially, because it's much faster to make the proteins. You're not waiting around to like grow a cell line up. So you add your DNA to what cell-free basically means. I grew up some cells, and then I popped them open, took their guts, put it in a tube, and then when I add DNA to it, remember high school biology, DNA, RNA, protein, it turns into protein. All right, and so you can change what else is in the mix. You can add endogenous extra chemicals to the mix that aren't just the guts of the cell. Depending on what you add, you get more protein out. The things you add cost money, but you know how much money they cost. So you could say a science project would be, what is the most optimal set of things you can add that give you the most protein for the least dollars? Okay, so protein per dollar produced. And that was what we set as the metric. We're like, GPT-5, your job is to put the right things in this mix that give us the most protein per dollar, all right? And the thing we got lucky on, we actually already chosen this as the project. And then Mike Jewett at Stanford, who's like the world's leading like cell-free, in my opinion, protein synthesis guy. Scientist, he published a paper that basically benchmarked a bunch of the different current mixes on price. And then his student did a ton of experiments, ultimately not as many as we did. We'll talk about that in a second. But and beat state of the art by 90%. And so there was a new state of the art as of August. And we're like, let's beat that. Okay, and then we ran the experiment. At first we let GPT-5 basically design with we only gave it access to like its own brain. So it didn't have a computer, it couldn't read the internet, only on whatever it had been trained on originally. All right, and this was actually GPT-5. We're on 5.4 now, but we started at the experiment, it was 5, and so we kept it 5 the whole time. All right, so we're like, okay, figure it out with what you know. And it designed experiment. Now we had a rule. This is actually kind of useful because like for doing AI for science, I think this is the biggest experimental AI for science result that has happened so far. Okay, and so I'll give you some tips and tricks for people that are interested in bringing AI into science. So one of the things we did was we had what's called a pedantic model, and this is open source, you can download it, our model, but it basically is a set of formal rules that in order for the model's experimental design to end up on the autonomous lab at Ginkgo, it has to pass these tests. So there's a 384 well plate. I can show you what one of these looks like, but there's like 384 little wells that you can add liquid to. So each one of those is an experiment. So a few things are in the rules. 1, you got to design 384. Don't waste any wells. 2, you can only add a certain volume of liquid. Otherwise it overflows. Or even at a certain level below overflowing, at the speed we move the things, it could slosh or something. So you have that as a rule, okay? So you set these rules. And also you need to reserve these 2 columns for controls, because we're gonna publish this and we're gonna need controls. And also you gotta do replicates. So each one of your designs, you gotta put it in 4 times in a row the same, so that we have replicates. So we set those rules. But then within those rules, it's all up to you, GPT-5. You could just tell it to fill it with water, and we'll fill it with water, okay? Whatever stupid idea you have, we'll try. Does that make sense? All right, so first off, we didn't even know, would it be able to design an experiment that like express protein at all, right? Like would it just do something crazy and ruin it or something, right? And so out of 500 of these 384 well plates that we ran, only 2 were like fill it with water, like where it really was like messed up. And partially, one of them, it was in part because we had the pedantic model, we missed something. And at one point, in order to try to get more reagents in, because we had that cap on volume, it added some reagents at a negative volume, which is, physically impossible, but hey, clever. And so we had to make a rule, you can't do it. And so like it messed up 2, but 498 plates, in my opinion, were fine experiments to run. In other words, they would contribute some data back that it would learn something from. Does that make sense? All right. So we let it do that. And then for 2 rounds, we said, you don't get any information. And so it started off, I forget what it was 10 or 20 times worse than the benchmark with the first ideas. It got a little better and a little better. But it wasn't like dropping like a rock, but it was improving. So it was able to like look at the last round of experiments, and then when it designed the next round, it was better, not worse. So that's already interesting, right? Like these are things that we weren't sure, right? And then we said, all right, well, let's let it have everything a normal scientist would be if I gave a project to. You could go read the literature. Here's Mike's paper from Stanford, tells you like the latest and greatest. Here's a computer. You want to write code, here's Python compiler, like go to town. And then on the very next design, it beat state of the art. Okay, and then in the next 2 designs after that, we were able to beat state of the art by 40%. So pretty neat. Now, a couple things were like unfair to Mike. We did more experiments than they did. So was it smarter, or did it just have more horsepower? Mike didn't have an autonomous lab. He had a really good grad student. They, right, who did a lot of experiments, right? But GPT-5 had access to robotics infrastructure. Right. Okay, so I think we don't yet know, like, is it better than a scientist at doing the same thing a scientist is doing? Sure. But we do know it can do the work of an experimental scientist, which we did not know before we started the experiment. Does that make sense? And we know it can do it at a level that's like, not even just like the beginning stuff where we showed, it can learn and do whatever, that it could read a current state-of-the-art paper and do the experiments that improve on the state of the art. Like that's a big deal. Now, this isn't like cure cancer, it wasn't the most creative work. This is the kind of work that you would describe a little more as like exploring an experimental space. But hell, that's a lot. Like back to my point earlier, I think the Einsteins are the rarity of science, who are sort of stewing and coming up with a theory and a breakthrough. The majority of science is testing the real world. And just being logical about what you see, and then being careful about the experiments you run, and then sharing your results. It did all that. So that means we can run these things Like if we want to think about the US running science, why aren't we spinning up 10,000 agents working on a particular science problem, all with access to autonomous labs, doing different experimental hypotheses, and work on it?